So, how is serum cholesterol regulated? So, we're often going to refer to terms as like fat flux or cholesterol flux, and that has to do with sort of this dynamic equilibrium, if you will, of taking something out of the bloodstream and putting it back in. We really didn't use this word when we talked about glucose flux. We talked about glucose homeostasis, but it's really the same thing. How are we thinking about how we're taking glucose out of the bloodstream when we've got high levels of blood sugar, or how do we put glucose back into the bloodstream when our blood sugar levels have fallen? So the same can be thought of with serum cholesterol is we have mechanisms for taking cholesterol out of the bloodstream in terms of taking up LDL particles. And then when we need to offload cholesterol from cells, when there's too much on the inside of cells, we'll talk about how we sort of efflux cholesterol. So there's sort of an influx and then an efflux, if you will, of sort of cholesterol particles that we have here. So we'll talk about this as an influx and an efflux really as an uptake and an export. So an import and an export. So first here, we've got three images here that we're going to talk about. First, I'm going to kind of highlight what happens in the normal sort of situation. And then we'll talk about an uptake sort of issue and an export issue that result in two different disease states. Okay, so normal situation here. Okay, normal situation is we are going to have uptake of LDL particles via receptor-mediated endocytosis. And we have another video up there about receptor-mediated endocytosis if you need a review of that. But this basically is going to bring LDL particles and their associated cholesterol into cells. Okay, so this uptake piece I'm going to highlight here, and then we'll sort of zoom in and we'll talk about the details of it. So that is this part that we have here. Okay, so zooming in here, we've talked about LDL particles. So this is an LDL particle here. I really like this picture because it reminds us that we've got this phospholipid monolayer, and then we literally have protein that's wrapped around the outside. This is this apolipoprotein B100. We call this ApoB100. That's the protein that I wanted you to remember that is wrapped around things like LDL particles, okay, which are going to take them out to the bloodstream, okay? Now, we're going to have these particles out in the bloodstream, and so now they need to be taken out of the bloodstream. We're going to uptake them into cells that's needed. So this is the receptor-mediated endocytosis. So if you remember from that video, we had the LDL receptors. They look like little Ys. Okay, so they're going to bind to the ApoB protein that's there. We're going to have those clathrin triskelions kind of help invaginate this. We're going to bring this particle in. Now, this is the only problem with this picture is it makes it seem like we only bring in one LDL particle. That is not necessarily the case. We're going to bring in lots of LDL particles, but one receptor is going to bring in one of those LDL particles. But this happens, we've got lots of particles that we're sort of bringing in. Ultimately, at the end of the day, we talked about how we have fusion with endosomes and lysosomes to ultimately release that cholesterol into the cytosol of that cell. And then we've got that cholesterol that's there. Now, that's going to be the influx or the uptake of cholesterol. Well, we also can have the biosynthesis of cholesterol in cells. And so cholesterol then can be elevated in its levels. And so we may need a mechanism to offload or export cholesterol. So that's what this next pathway is. Okay, so I'll talk about it since we've got the picture right here, and then we'll put it into words. Okay, when you have excess levels of cholesterol, what's going to be happening is you're going to use this transporter system called ABCA1. Okay, this is going to be a transporter system that essentially recruits HDL particles. It's going to be interacting with the apolipoprotein A1. So we talked about ApoA1 is on HDL, ApoB100 is on LDL. Okay, so these HDL particles are going to dock there. They're going to take in cholesterol that needs to be offloaded or exported from cells. And then that ApoA1 protein, okay, that's going to be what directs those HDL particles back to the liver. And remember, the liver is the only place where we can dispose of excess cholesterol because excess cholesterol can be produced into bile acids. Bile acids can be put into your GI tract, and that is the only way to rid your body of excess cholesterol. Okay, so this now is going to be our export sort of pathway. So I'm just going to highlight this in green here, and then we'll come up top and sort of talk about what's happening now in words, okay, when we have this export portion. So the export is what we just highlighted in green, okay? And so this is going to be via the ABCA1 sort of transporter. Okay, so you're going to display this on your surface. Okay, so you're going to display that you've got, excess cholesterol. It's going to be a signal that you've got excess cholesterol, and HDL is going to come by. It's going to dock. It's going to pick up that cholesterol, take it to the liver, again, export it via bile acids. So HDL is going to, let's write dock, because that's what happens. It docks and picks up that cholesterol, and it's going to take it to the liver. If it's truly excess, the liver can make it into bile acids, and then you can dispose of it in fecal material. Okay, so that's what happens in a normal situation. So the next two sort of pictures here are going to highlight what happens in two situations that lead to disease states. So the first one that we're going to talk about is called familial hypercholesterolemia, often just abbreviated FH. Right, let's break down this word here, right? Hyper, high levels, cholesterol, high levels of cholesterol, emia, in the blood. So high levels of cholesterol in the blood. And if we just look at this picture, we can see what's missing, okay? We do not have LDL receptors on the surface. And so what's going to happen here is we can either have missing LDL receptors, so maybe there's an inadequate synthesis of LDL receptors, or maybe you have that faulty recycling of those LDL receptors back to the surface. Both of those ultimately are going to increase the concentration of LDL particles in your blood, and this can lead to LDL cholesterol concentrations in the magnitude of 300 to 500 milligrams per deciliter, right? Much, much larger than we would expect for sort of normal serum LDL levels. Okay, so again, this familial hypercholesterolemia, it's generally from missing or faulty recycling of those LDL receptors. Okay, and what this is going to lead to is elevated levels of LDL in the blood. And again, we see things on order of 300 to 500 milligrams per deciliter. So on the order of about 3 to 5 times higher than we would expect. Now, again, that can be problematic. We're going to get into the details of this later on in the course. But when you have sort of endothelial dysfunction in your vasculature, and you've got not, a very smooth endothelial lining there, you've got pits in it, you've got sites of inflammation, things like that, that can cause these LDL particles to stick. And that can sort of lead to soft plaque formation, which can lead to calcification, ultimately leading to, atherosclerosis and, problems with cardiovascular disease. All right, so familial hypercholesterolemia, a genetic component to it, as is indicated by the familial piece, right? But that is generally an issue because there's a problem with the uptake of LDL from your bloodstream into your cells, okay? Either missing receptors, faulty recycling. Okay, this last one here is a little bit different, and I'm going to highlight that this is called Tangier's disease. Most of you are not going to worry about having issues with Tangier's disease. We're going to talk about sort of the pathophysiology behind it, but in reality, there's only about 100 people in the world who have Tangier's disease, and this is again a disease of the export piece. So highlighting here, let's just go in and put this in red, that when we have this missing or faulty LDL recycling of the receptors, that's an uptake issue. That's why we have, cholesterol issues. So familial hypercholesterolemia is really an uptake issue. Tangier's disease is really an export issue. So again, if we see what's happening here, we have no problem sort of bringing in cholesterol from the bloodstream, okay? We also have the potential of, producing our own cholesterol, right? But that's not likely to be the case when we have such high levels of cholesterol on the inside. But if you're constantly having, if we're talking about this flux sort of control, we've got influx and efflux. If we're constantly having an influx but no efflux, that's going to cause cholesterol to build up inside the cell. So the key with this Tangier's disease is there is no export mechanism. So no export mechanism. Okay, and so we're going to have accumulation of cholesterol in the cells. Ultimately, this can recruit macrophages. This can be very inflammatory. There's lots of neurological sort of complications here. This is not something where you tend to see, the elevated LDL cholesterol particles as really being the problem here. It's really this accumulation of cholesterol within cells because you cannot offload it. Actually, one of the hallmarks, and actually how was it identified the first time, and I think this was in like the 1950s, is there was a 5-year-old girl that was on Tangier's Island, and they noticed that her tonsils were like yellow, orangish in color, and that's because they were just full of cholesterol because there was no way to sort of offload it from cells. Okay, so this can be a very inflammatory problem. We can again recruit macrophages. A lot of times when there's too much of a metabolite or a biological molecule that's within cells, that's an indication that, hey, macrophages, come on along, like destroy and get rid of this cell. There's something problematic with it. But again, if it's all the cells that are in your body that's problematic with, rather than just a rogue 1 or 2 cells, then this can be problematic sort of from a disease state. But again, highlighting that this is about 100 people in the world. It's autosomal recessive, so it's not something that kind of tends to propagate in a population. There's no selective advantage for it. It's sort of been isolated to communities within this geographical region. So cholesterol flux, really, really important to maintaining sort of healthy levels of cholesterol in your bloodstream.